Thank you for joining me for today's presentation. My name is Julio Camperio, and I'm currently an aquaculture PhD candidate at the University of Miami in Florida, focusing on fish nutrition. I understand that the conference was delayed a couple of times due to COVID-19, and technically I should be in Germany in person presenting this research to you. But due to traveling bans and restrictions, I'll be presenting this project remotely. Before we begin, I'd like to thank the organizers and participants of the conference for selecting and awarding me the Muchman Award. This is a true honor for me, and I gladly and proudly accept it. Nevertheless, this award is also being given to all the people and organizations that assisted in this research project. Without them, this would not have been possible. This presentation is a accumulation of 3 different experiments geared towards the same issue of amphibian captive rearing. That being said, the topic of this presentation is Chytridiomycosis, Spindly Leg Syndrome, and water composition in amphibians. Let's begin with Chytridiomycosis, also known as chytrid. Chytrid is an infectious disease caused by the aquatic fungal pathogen Batrachochytrium dendrobatidis, also known as BD. BD has caused global declines or extinctions of at least 90 species of amphibians. Some of these drastic declines or extinctions occurred within 1 year species infection and even occurred in remote and pristine habitats. There are another 124 species that have seen their populations decline by 90% or more. The alarming aspect of this fungus is that it is the first emerging disease to have caused the decline or extinction of hundreds of frog species that were not otherwise threatened. Globally, this fungus has infected more than 500 species of amphibians. On the image on the left, you can see the global spread of BD across multiple continents, which is clearly an alarming fact. The effect of chytrid on juveniles and adults is to insist within the skin cells, which with time become thicker and eventually sloughs off or peels off. Osmotic regulation is increasingly compromised and electrolyte blood levels drop, leading to death from cardiac arrest. Chytrid seems to have an Asiatic origin with an incredible rate of expansion. A possible reason for this expansion is due to human-related amphibian trade for consumption, pet trade, research, bait, and accidental transportation. Another possible explanation for the rapid expansion of the fungus is due to climate change, which forces species to migrate and thus spread the fungus. In terms of the genus Atelopus, which is what the research presented today will focus on, of 113 described species, at least 30 species appear to be extinct. Only 52 out of the 83 surviving species have enough data to evaluate population trends, which indicate that around 80% of these species have been reduced by at least half. On the image on the right, we can see the chytrid life cycle on how it develops and infects more frogs. The fungus and the ensuing disease are indeed a critical aspect in amphibian conservation. At the moment, no cure exists for chytrid. In captive animals, BD can be treated with increased temperature or antifungal treatments. In wild animals and in wild environments, we sadly do not have the tools to successfully control BD, which greatly hinders reintroduction efforts. Another method to fight BD is the use of bioaugmentation. Some surviving populations of Atelopus varius have evolved anti-BD skin secretions, and scientists at PARC are actively working to selectively breed for resistance and to identify resistant genotypes. Improving sustainability of captive breeding collections is a major research area for the Panama Amphibian Rescue and Conservation Project. Scientists and conservationists need a healthy and steady supply of captive bred frogs to study and research BD, and to develop new methods for counteracting it. This last point is very important because it's where Spindly Leg Syndrome, the main topic of today's presentation, comes into play. Before we move on, I wanted to turn your attention on the image on the upper left, which is one of the reintroduction methods using tracking devices on an Atelopus ziteki. The 2 images on the bottom are the method used to detect either BD or the anti-BD skin secretion on some of the surviving populations. Spindly Leg Syndrome, also known as SLS, is a musculoskeletal abnormality in captive reared amphibians, resulting in underdeveloped front limbs. On the left, you can see images of frogs that have SLS, and notice the underdevelopment of the front limbs. If SLS occurs, the frog will not be able to move and will thus not be able to eat, eventually leading to mortality. The cause of SLS has not been fully pinpointed yet, but it is to be believed that it's either a nutrition deficiency in larval diets, mainly vitamin B, husbandry factors, mainly water quality, or be a multifactorial cause such as maternity and genetics. What is known is that metabolic bone diseases are linked to improper regulation of calcium metabolism related to poor dietary calcium phosphate ratios, vitamin D, or inadequate UVB exposure. Going more into depth on this research that was carried out on SLS, we'll take a quick look at the Panama Amphibian Rescue and Conservation Project. This project and its facility was created in 2009 to carry out the ex situ assurance population of amphibians in Panama at risk of extinction from chytrid. The occurrence of SLS has been observed in Atelopus limosus, Certus, Glyphus, Adenobates heminicea, Ophaga vicenti, and Hylocertus colimba. The prevalence of SLS introduces uncertainty for population managers who see a large portion of their captive reared frogs not making it to adulthood, which greatly reduce the individual number of specimens that they can work with. Additionally, it is a research priority for animal husbandry and well-being of captive Atelopus to maintain a healthy and stable captive population. The two images on the bottom are photos of the actual conservation center. What is interesting of this facility is that species that belong to similar environments have been placed in shipping containers, which have been retrofitted to become isolated and contained rearing rooms. The facility, I believe, has 4 of these containers. In the image on the top right, we have Roberto Ibanez, who is the director of the Panama Amphibian Rescue and Conservation Project. Regarding SLS, 3 experiments were carried out. The first one looked at the influence of dietary protein content on the prevalence of SLS. The second experiment looked at the influence of water composition, ration, and diet type on the prevalence of SLS. And the third experiment looked at the environmental calcium and phosphate availability on the prevalence of Spindly Leg Syndrome. I carried out the second experiment. The first one was carried out at an earlier date and also included in the same publication, while the third experiment was carried out on a later date. Experiment 1 hypothesized that the lack of dietary protein in Atelopus tadpoles causes SLS. This was based on anecdotal observation that by increasing animal protein in Mantella aurantiaca tadpole diets, the prevalence of SLS is reduced. Experiment 1 was carried out on 400 tadpoles of Atelopus certus at Gosner stage 22. There were 16 tanks with 25 tadpoles per tank. Each tank had about 2.5 centimeters of gravel with an undergravel filter and airstone. Tanks had a total capacity of 20 liters but were filled to 16 liters with water held between 22.3 and 24.1 degrees Celsius and were provided 12 hours of light with a bi-weekly water exchange of 20%. There was a total of 4 treatments consisting of isocaloric diets with 31, 37, 42, and 48 crude protein. This was based on surrogate frog species diets and natural food samples collected from the wild. Each tank was provided with 2 acrylic feeding plates and floating polystyrene for morphing tadpoles. This experiment was carried out to quantify SLS proportion, survival through metamorphosis, and days for surviving tadpoles to metamorphose. In terms of results for experiment 1, all diet treatments produced more than 80% of SLS. All treatments experienced average mortalities of 30 to 50%. Dietary protein levels seem to not be a primary cause of SLS. Additionally, high levels of vitamin B and supply of UVB light on all treatments exclude these two as limiting factors. Experiment 2 was conducted to test 3 different hypotheses. The first one is that by reducing food quantity, SLS will be reduced. This was based on anecdotal observation that by reducing food quantity of Atelopus tadpoles, metamorphosis occurs earlier and with less SLS. The second hypothesis is that some unknown nutritional factor may cause SLS. This is based on reports that Atelopus tadpoles fed on a commercial diet recommended by Species Survival Program had no SLS. The third hypothesis is that reverse osmosis treated water will reduce SLS. This is based on observation at a sister facility that treated water will indeed reduce SLS. Experiment 2 was carried out on 720 Atelopus glyphus tadpoles at Gosner stage 22. The experiment consisted of 36 tanks with 20 tadpoles per tank. Each tank was provided with a sponge filter, an air stone, 2 acrylic feeding plates, and a PVC fitting. The total capacity of the tanks was 30 liters, but each was filled with 26 liters of water and it was kept between 22.3 to 22.5 degrees Celsius. There was a tri-weekly water exchange of 30%. Regarding treatments, we had about half of the tanks with reverse osmosis water and the other half with tap water. Of the tanks with reverse osmosis water, we provided either a full, half, or quarter ration of experimental feed, half a ration of commercial feed, or half a ration of commercial feed with supplemental vitamin B. Of the tanks with tap water, we provided either a full, half, or quarter ration of experimental feed, or half a ration of commercial feed. Each tank was also provided with a floating polystyrene for morphing tadpoles. This experiment was carried out to quantify SLS proportion, survival through metamorphosis, and days for surviving tadpoles to metamorphose. In the image on the top left, we have a glyphus tadpole laying by the acrylic feeding plate, and in the background, you can see the air stone and the PVC fitting. In the image on the top right is the room where the experiment was carried out. We have the table where the feed was prepared daily on the far right. The black rack on the right is where we kept the 36 tadpole tanks, and the rack on the left is where we kept the metamorphosed tadpoles. Each tadpole tank was assigned its own froglet container. And on the far left, we have the reverse osmosis system that we used to treat the water. And on the image on the bottom right is a close-up of a tadpole feeding on the actual acrylic plate. In terms of results for experiment 2, diet type did not significantly affect the prevalence of SLS. Water source and ration did significantly affect SLS incidence. And looking more specifically to tap water, the incidence of SLS and ration were correlated. This might be due to higher ration causing biofouling, which may have negatively affected water quality. Post-metamorph morphality of RO was half of that of tap water, with 27% compared to 57%. Diet type significantly affected tadpole mortality. Ration significantly affected tadpole mortality, with a higher mortality with the commercial diet and full ration. Additionally, it took the full ration treatment 15 to 20 more days to metamorphose. Experiment 3 was carried out to test 2 hypotheses. The first one is that available environmental calcium will affect SLS incidence. This is based on studies that soft water was related to skeletal deformities in tadpoles and metamorphs. The second hypothesis is that phosphates released from uneaten food might upset the calcium-phosphate ratio and aggravate SLS. This is based on studies that overfeeding tadpoles causes higher incidence of SLS. Experiment 3 was carried out on 600 tadpoles of Atelopus varius. The experiment consisted of 30 tanks with 20 tadpoles per tank. Each tank was provided with a sponge filter, an air stone, and 2 acrylic feeding plates. The total capacity of the tanks was 30 liters, but they were filled to 26 liters with filtered reverse osmosis water, which was then reconstituted. Water temperature was kept between 22.3 to 22.5 degrees Celsius. There was a tri-weekly water exchange of 30%. There were 2 main treatments, low phosphate and high phosphate. Of both phosphate treatments, we had an additional sub-treatments of low, medium, and high calcium. All tanks were fed 0.125 grams of nutritionally balanced food. Each tank was provided with floating polystyrene for morphing tadpoles. This experiment was carried out to quantify SLS proportion, survival through metamorphosis, and snout vent length and weight of metamorphs. In terms of results of experiment 3, a calcium level of 50 milligrams per liter significantly improved survival. 4 of the treatments that had an additional of calcium had 49% survival versus the other 2 treatments without calcium supplementation only had 19% survival. The addition of phosphate and interaction of calcium and phosphate had no effect on survival. High and medium calcium had 15% of SLS incidence versus 60% with low calcium. In terms of calcium, it appears that 50 milligrams per liter is the threshold. In the low calcium treatments, the addition of phosphate reduced SLS incidence. In terms of phosphate addition, there was a 23% SLS incidence with phosphate as compared to 39% SLS without phosphate. There is no significant difference in size, weight, and days to metamorphose. Overall, it seems that with experiment 1, the protein content was not a significant factor in prevalence of SLS. In experiment 2, the conclusion was that protein quantity or quality or the addition of vitamin B are not key factors. In fact, it's the water composition that seems to be the main factor in SLS prevalence. Additionally, as all husbandry, it is important to avoid overfeeding as it might have unaccounted effects on water chemistry. In terms of experiment 3, the overall conclusions are that the addition of calcium significantly improves survival. If the access to reverse osmosis water is not possible, then supplementing the water with calcium chloride might be an effective alternative. If the water is softer than 50 milligrams per liter of calcium hardness, you can supplement with calcium chloride. In conclusion, I would like to thank the following organizations and people that have made this project possible. Without them, these experiments might not have materialized. I would like to thank all of you for your time and attention, and I'll be available to answer any questions.
So in terms of questions and answers, here's a following question that I was given. Why do you still see 15% SLS with a sufficient amount of calcium? We don't have enough experimental evidence for why the SLS persists even with sufficient calcium, but we hypothesize that it is connected to overfeeding of the tadpoles as it is explained in this publication. Tadpoles absorb calcium across their gills and store it in endolymphatic sacs in preparation for metamorphosis. If they're growing faster, meaning that they're absorbing calories across the gut, faster than they accumulate calcium store in preparation for metamorphosis, this could lead to a decoupled growth and calcium sequestration. An experimental approach that limits calories to an extent that significantly delays metamorphosis in comparison to a non-calorie limit group under conditions with suboptimal calcium levels to actually induce a reasonable response frequency of SLS in both groups would be required to test this. We would recommend also examining and measuring calcium storage in the endolymphatic sacs as a response variable in a sacrificial design to test this hypothesis. Atelopus has been a good module system for use as a single clutch of eggs allows for adequate replication compared to dart frogs, which have been more challenging. Our take-home message for husbandry people is faster is not always better. Don't rush and take it slow. Give the tadpoles time to grow strong rather than fast. For the second question, can you say which water values you created and what kind of mixture between tap water and reverse osmosis water we used, and how we reconstituted the water? We did not mix tap water and reverse osmosis for any treatments. In fact, because our tap water is so soft to begin with, our current policy is to use carbon-filtered tap water in all tadpole tanks, and simply to add calcium chloride and magnesium sulfate as if we were to reconstitute reverse osmosis. Which additive do you use to enhance the mineral content of the osmosis water? Preparations for a fish aquarium? We've used a variety of products for mineral reconstitution, ranging from lab-grade chemicals purchased from Sigma to more cost-effective food-grade chemicals purchased online that are readily available from multiple suppliers. So really cost, size, and shipping speed are the factors that we take into account more than the brand. We use a lot of calcium chloride and use Dr. Foster Smith formulation for reef tanks because you can easily buy it in large amounts and it's not food grade, but it is meant for aquarium end users. What was the recipe for the minerals added to the reverse osmosis water? To reconstitute the reverse osmosis water, we dissolved 0.0395 grams of calcium chloride, 0.0465 grams of magnesium sulfate, 0.0358 grams of potassium bicarbonate, and 0.0298 grams of sodium bicarbonate per liter of reverse osmosis water. Here we have a list of the water quality analysis of tap water, reverse osmosis water, and the reverse osmosis water with the added salts. The way we had the system set up was as follows. Tap water was plumbed to a series of mechanical and carbon pre-filters, then to the RO filtration system. RO water drained into a 90-liter reservoir with a float valve stop. On water change days, reverse osmosis water was pumped from here to a secondary mixing reservoir, also 90 liters. The salts for reconstitution were added to the second reservoir to the appropriate amount. This reconstituted water was then used for manual water changes. Did you compare your water to the habitat waters that tadpoles lived in? The water values and calcium, for example? Yes, these values are presented as supplementary material in this paper. The stream water was also very soft. This is why we suggest further research into the potential dysregulation between calcium sequestration and calorie input. We don't know the natural wild development times of Atelopus tadpoles. Can you tell us the composition of tadpole feed that was used during your study? Was it a gel mix applied to acrylic plates? All feeds came in a dry powder form. All dietary treatments were first weighed according to feed ration, then mixed with water and turned into a paste. The paste was then spread onto 2 acrylic plates per tank. Note that the plates were scratched with sandpaper to make the surface more coarse and allow the feed paste to better stick. The plates were then allowed to dry for 24 hours. Here, as you can see, is the nutritional profile of the feeds that were used in the experiment. Were there any heavy metals, herbicides, or pesticides in tap water that are also relevant, as they are also filtered out in osmosis water? We initially suspected some unknown water quality contaminant, which is why we conducted an RO treatment in a second experiment, which should also reduce contaminants considerably. When we conducted a full spectrum elemental analysis on both tap water and RO water, we found that there were no abnormally high values for heavy metals, but there was very low values in particular for calcium in the tap water. We therefore suspected calcium to be the limiting factor and ruled out contamination. Given our knowledge of calcium phosphate rations and gut loading guidelines in adult frog diets, we suspected that phosphate in the water may have been high relative to the calcium. As such, the final experiment only used RO water and tried to control both calcium and phosphate. We were a little surprised to find that calcium and phosphate were both limiting in our system.